Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be reviewing for Exam 3 for Biology 1408, Biology for Non-Science Majors. Let's go through Chapter 7 first. So here, Chapter 7, we are covering cellular respiration and fermentation. I want you to know what cellular respiration is. Remember, cellular respiration is the process of converting glucose, you can see here glucose, oxygen, into CO2, water, and ATP. So the reactants are glucose. You're starting with glucose, which is C6H12O6, and you're starting with oxygen, O2, and you are forming what? You are forming CO2, you are forming H2O, and you are forming some ATP, okay? So you should know the reactants, glucose and oxygen, and the products, CO2, water, ATP. And where does cellular respiration occur? You should know that cellular respiration occurs in the mitochondria. The mitochondria, right? All right. What else do we need to know? I'd like you to know about, look at this slide here. These are the stages of cellular respiration. Stage 1, glycolysis. Stage 1.5, oxidation of pyruvate. Stage 2, the citric acid cycle. And remember, the citric acid cycle is also called the Krebs cycle, and it's also called the TCA cycle. And then you have stage 3, oxidative phosphorylation. One thing I want you to know is that stage 1, glycolysis, occurs in the cytoplasma, right? Or the cytoplasm of the cell. So you should know that glycolysis occurs in the cytoplasm of the cell. Whereas you should know that the rest of these stages, stage 1.5, stage 2, and stage 3, they occur in the mitochondria, right? So please know that glycolysis occurs in the cytoplasm of the cell, whereas the rest of cellular respiration occurs in the mitochondria. Now, this is a great reference slide for you. This is a great reference slide because it's showing you how glucose is converted to, the energy in glucose is being ultimately converted to ATP. During glycolysis, glucose is broken down into 2 pyruvate, also 2 NADH are formed, and 2 ATP are formed. So let's talk about glycolysis real quick. Remember, glycolysis occurs where? Glycolysis occurs in the cytoplasm. And what does the equation look like? You start with glucose, and during glycolysis, the products are 2 pyruvate, 2 ATP, and 2 NADH. So again, I want you to know what things about glycolysis. I want you to know that it occurs in the cytoplasm. You start with glucose, and what are the products? 2 pyruvate, 2 ATP, and 2 NADH. So again, you should know that you start with glucose, and what are the products of glycolysis? Please know the products of glycolysis. Excellent. Now, this is step 1.5. This is step 1.5. Remember, this is called oxidation of pyruvate. So during step 1.5, oxidation of pyruvate, again, I'll show you what I'm talking about real quick. You see, stage 1.5 oxidation of pyruvate. What do we need to know about that? Let me show you. So remember during this step in the lecture videos, we're starting with 2 pyruvates. Actually, everything here is multiplied by 2. 2 pyruvates are entering the mitochondria. And when the 2 pyruvates enter the mitochondria, you have oxidation of pyruvate. So when that happens, when you oxidize pyruvate, you have CO2 formed, you have NADH formed, and you have acetyl-CoA formed. Did you follow that? So again, during stage 1.5, you start with the reactant pyruvate. We're starting with pyruvate, and we are ending up with what? The products are CO2 and NADH and what? Acetyl-CoA. By the way, if you ever want a quick reference for any of these stages of cellular respiration, just go back to this slide. Let me show you this. This overview slide is wonderful because look at this. Remember glycolysis? You start with glucose, and what were the products? NADH, pyruvate, and ATP. And this is stage 1.5. Remember stage 1.5, which we just touched on, which is called oxidation of pyruvate? What were the products we just talked about? NADH, CO2, and acetyl-CoA, right? So what's the next stage? It looks like the citric acid cycle or Krebs cycle, also known as Krebs cycle, also known as TCA cycle. That's the next stage. So now we're going to talk about this citric acid cycle. You know what I mean? We're going to talk about stage 2, the citric acid cycle. So let's talk about stage 2. And remember, this is just a review right now. If you want full details on what all this means, remember we have those full-length lecture videos for you to watch as well, so you can see all the little details about what all is going on during the citric acid cycle. That's in the lecture videos. This is just a review for the exam. I want to highlight the important things that you should know so you can do really well on the exam. That's the point of this review here, not to explain the citric acid cycle because that's in a different video, but just to highlight the important things from those lecture videos, right, for the exam that you should really focus on for the exam. Okay. So this is the citric acid cycle. Remember, it's also known as the Krebs cycle, which is also known as the TCA cycle. So what's the reactant here? It looks like the reactant is acetyl-CoA. So we're starting with acetyl-CoA. This is stage 2. And what do we produce? What are the products of the citric acid cycle? Well, it looks like here, look, we're making NADH here, here, and here. We're making NADH. What else are we making? Our products are CO2. We're making CO2. Now, you don't need to know the exact numbers of these things, but we're making CO2. So we've made NADH. We've made CO2. What else do we make? Looks like we also make some ATP, right? ATP. Make some ATP. And what else? There's one last product. Yes, check it out. There's FADH2. I want you to know FADH2. These are the products of citric acid cycle. Okay, let me show you again this overview slide I told you about that you should refer to. Remember, what did we just say? During the citric acid cycle, acetyl-CoA comes in, that's the reactant, and what do we make? We make NADH, we produce FADH2, we produce CO2 and ATP. Okay, wonderful. So that just leads us to the final stage. Remember, the final stage is stage 3. This is called oxidative phosphorylation. Let's go through and highlight what you need to know about that. All right, let's go to stage 3. All right, this is stage 3. This is oxidative phosphorylation, stage 3. Okay, so what all is going on here? What I want you to know is that for stage 3, this is all I want you to know about stage 3, the oxidative phosphorylation. I want you to know that you're using your NADH, so the reactants are NADH, and you're using FADH2, and what are you producing here? What are you producing? Oh, and you're also using oxygen. Sorry, you're also using oxygen. Remember, oxygen is what? Oxygen is called your terminal electron acceptor. Oxygen is your terminal electron acceptor. So again, what do I want you to know about stage 3, oxidative phosphorylation? I want you to know that it requires NADH, it requires FADH2, and it requires oxygen, the oxygen that you breathed in. And what are the products? What are the products? The products are ATP, and what else? And water. And again, if you want to have this this process at a glance, you know, the reactants and products of this process at a glance, let's go back to our cheat sheet. Remember our little cheat sheet? This is our little cheat sheet. Remember, during stage 3, oxidative phosphorylation, it requires NADH, it requires FADH2, it requires oxygen as the terminal electron acceptor. And what do we form? We form ATP and we form water. Awesome. So those are the things that we really want to know for the exam. Okay, those are the main things. And I've made a note here of these main things that we just reviewed that you should know for the exam. Okay, those are the things to know for the exam. As far as fermentation goes, what do I want you to know? As far as fermentation goes, what I want you to know is simply this. We don't need to go too much into fermentation for 1408. But what I do want you to know is that your body switches from cellular respiration to fermentation when? When does your body switch from cellular respiration to fermentation? When does your body start making lactic acid? Do you know? Remember from the lecture videos, this is when you are low on oxygen. When you aren't providing your tissues with enough oxygen, let's say you're working too hard at the gym, you know, this is when your oxygen levels are too low and your body switches from cellular respiration to fermentation. And all I want you to know is that, again, fermentation occurs when we are not receiving enough oxygen, and so we are making much less ATP. How much ATP do we make during fermentation? We are making only 2 ATP, okay, net, 2 ATP. So again, all I want you to know about fermentation for the exam is that your tissues switch to fermentation when there's not enough oxygen, the terminal electron acceptor, and then your body only can make 2 ATP per glucose instead of the normal 30 net ATP per glucose. Okay, so that's about it for this chapter. That leads us to the end of this chapter. I made a note of the things that I just covered with you about chapter 7. I know it's a challenging chapter, but like I said, focus on the details that I just outlined for the exam. I think you'll do just fine. And let's, after we take a little break time with Gizmo and Wicket, let's hop into chapter 8, make sure we're prepared for the exam there. Hey everyone, welcome back from our break time with Gizmo and Wicket. Let's continue on with review for Exam 3, Biology 1408, with Chapter 8, Photosynthesis. I want you to know the concept of photosynthesis is the process that converts solar energy into chemical energy in the form of sugar, right? Chemical energy in the form of sugar. And where does photosynthesis occur? I want you to know that it occurs in the chloroplast. Remember the chloroplasts? It's the photosynthetic organelle. I want you to know that the creature is called autotrophs. These are the photosynthesizers. These are the ones that can sustain themselves without eating other organisms. So plants are autotrophs, whereas you and I, animals, are heterotrophs. We obtain our organic material from other organisms by eating them, okay? So humans and other animals are heterotrophs. Plants and photosynthesizers are autotrophs. So please know the difference between autotrophs and heterotrophs. And remember this, the autotrophs include plants, algae, these photosynthetic protists, as well as photosynthetic bacteria. What else? We should know the structure of a chloroplast. Do you remember how the chloroplast works? We have the outer membrane, the inner membrane. Remember what the structure of the chloroplast is? Remember these stacks inside of the chloroplast are called granum. Granum. The stacks are called granum. And each individual pancake, I call it, each individual pancake in the stack is called what? It's called a thylakoid. So what you should know is that this is a stack called a granum. Each component of the stack, each pancake in the stack, if you will, is called a thylakoid. The membrane of this stack is called a thylakoid membrane. This is called the thylakoid membrane. The fluid outside of the stacks is called the stroma, and the fluid inside of these stacks is called the thylakoid space. So let's know that for the exam. Now don't mix up stroma with stomata. Remember what stomata are? Stomata are these pores on the undersides of leaves that allow for gas exchange. And remember, plants take up mostly CO2 and they expel mostly oxygen, right? So the stomata are for gas exchange into the plant leaf, but the stroma is the fluid outside of these pancake stacks of the thylakoid, of the chloroplast. What else do we want to know? So again, the parts, we want to know the parts of a chloroplast. We want to know the difference between stomata and stroma. We want to know this. We definitely want to know this. This is the equation for photosynthesis. So you should know that photosynthesis, the reactants, the things you're starting with are light, CO2, and water. And what are your products? Your products are sugar and oxygen. Okay, so we want to know the equation for photosynthesis. What else do we want to know about photosynthesis? We want to know this. If I ask you, what are the steps, what are the major components of photosynthesis, you should know that there are two major components to photosynthesis, two major stages, right? Remember, the first component or stage is called the light reactions. And then the second component or stage is called the Calvin cycle. Now, you should know what are the reactants and products of the light reactions, and you should know what are the reactants and products of the Calvin cycle. So let's cover that together. The light reactions, see here, the light reactions, which occurs in the thylakoid. You should know the light reactions occur in the thylakoid. Light reactions occur in thylakoid. And what's the process here? Water is a reactant. Water is a reactant, and that water is split into oxygen, right? Because water donates its electrons. Oxygen is a product. NADPH is a product, and ATP is a product. And what else is required for the light reactions? Light. Okay, so what do I want you to know? I want you to know, let me show you. I want you to know that in the light reactions, let's talk about the light reactions. Your reactants are light, okay? Light. What else do you use? Water, right? Light and water are the reactants. And what are your three products? What are the three products of the light reactions? Well, right here, oxygen, oxygen, what else? NADPH, okay, NADPH, what else? And ATP, okay, ATP, great. That's what I want you to know about the light reactions. And remember, I'm not going through details on this review video. This is simply a review of the full-length lecture videos. But I'm just trying to highlight those important things that you should know to do well on the exam, so you can make sure that you're studying and focusing on some of the more important aspects, right, for the exam. Now, what's the other step? Remember, the other stage was the Calvin cycle. So what happens during the Calvin cycle? During the Calvin cycle, it requires CO2, ATP, and NADPH. These are the reactants. Did you see that? The reactants are CO2. So during the Calvin cycle, you start with CO2, you start with ATP, and you start with NADPH. And what are the products? What do you make? You make sugar. You make sugar. Yes, you form sugar. So the product is glucose or sugar. Glucose or sugar. And where does the Calvin cycle occur? Does it occur in the thylakoid like the light reactions do? The answer is no. It occurs in the stroma. I want you to know that the Calvin cycle occurs in the stroma. Very good. All right, let's keep going. So again, this is just an overview. This is your cheat sheet, if you will, for what happens during photosynthesis. You see, it's all the data you need is right here. All the information you need is right here. See, during the light reactions, remember, that's stage 1, and then the Calvin cycle is stage 2. This is all happening inside of a chloroplast. Remember the chloroplast? The pancake stacks? Okay, so the light reactions are occurring in the thylakoid, the pancake stacks. Remember what we require for the light reactions? Light and water. And what are the products of the light reactions? ATP, NADPH, and oxygen. That's what I want you to know. And then where does the Calvin cycle occur? The Calvin cycle occurs in the stroma. You know, remember the fluid outside of those pancake stacks? And what happens during Calvin cycle? You're taking CO2, you're taking ATP and NADPH, those are your reactants, and you're forming which product? Sugar. Usually it's glucose sugar, okay? So that's what we want to know. We also touched a little bit on light. Okay, we touched on light. Remember visible light? It includes the electromagnetic spectrum from about 380 nanometers to 750 nanometers. Red light is about 700 nanometers. Blue light is around 500 nanometers. Purple light's around 380 nanometers. What do we need to know about light? A couple of things. I want you to know that shorter wavelength light, so like purple light and blue light, has higher energy. So shorter wavelength light has more energy. More energy. That means longer wavelength light, like red light or orange light or yellow light. This has lower energy. What else do I want you to know? I want you to know that plants appear green because they are not absorbing green light, okay? So what you're seeing when you see a green plant or a green tree or green grass, what you're seeing is reflected light or transmitted light, okay? This is what the reason plants appear green is because green light is transmitted. That means green light goes right through them, and green light is reflected. But green light is not absorbed. I want you to know that green light is not absorbed by plants, or more specifically by chloroplasts. Even more specifically by chlorophyll. Remember chlorophyll? Chlorophyll is the photosynthetic pigment. So again, why do trees appear green? Well, because they're not absorbing green light. They're reflecting green light towards your eye. They're transmitting green light towards your eye, but they're not absorbing green light. So I want you to know that they're not absorbing green light. Okay. These slides here are a little bit more in depth, a little bit more for biology 1406. Okay, so I kind of, I think that's where I'm going to draw it for 1408 for you guys. That's the level of detail I want you to know. Those concepts we just touched on. All right, so I think that's doable. I think you can do it. Let's take another little short time, break time with Gizmo and Wicket, and we'll come back with our final chapter, chapter 10, so that we are well prepared for the third exam. What do you guys think? Hey everyone, welcome back from break time with Gizmo and Wicket. Let's proceed with chapter 10 of our exam 3 review for biology 1408. Obviously, please know the definition of the cell cycle. So know what the cell cycle is. Here's the definition for you. We want to know the difference between somatic cells and gametes. Remember, somatic cells are the non-reproductive cells that have two homologous sets of chromosomes. These are basically any cell in your body that is not the gametes, is not the sperm or the egg. The gametes are the other type of cell in your body. These are your reproductive cells, the sperm or eggs. They have half as many total chromosomes as somatic cells because they are haploid. Remember that term, haploid. Now, so we want to know the difference between somatic cells and gamete cells. All right, remember that term haploid and diploid? Do you remember? We need to know the difference between the term diploid and haploid. Diploid cells, these are your somatic cells. Your somatic cells are diploid. Diploid means they have two complete homologous sets of chromosomes. For example, humans have 46 chromosomes. One set of 23 they inherited from their dad, and one set of 23 they inherited from their mom. And these are known as the homologous sets of chromosomes. Your sperm or your egg, on the other hand, the gametes are the haploid cells. The haploid cells only have one set of chromosomes, so only 23 chromosomes. So please know the difference between diploid and haploid. Remember, your somatic cells are diploid and your gamete cells are haploid. What else do we need to know? we need to know this, the difference between sister chromatid pairs and homologous chromosomes, homologous chromosome pairs. There's a nice video here in your notes. If you go to your handouts, your notes, your lecture handouts, and click on this icon here at the bottom left, this is a nice overview of the difference between homologous chromosomes and sister chromatid pairs. So please know the difference. And just to review real quick, homologous chromosomes, that's like the blue chromosome and the red chromosome. Homologous chromosomes are not identical. They share the same types of genes. They share the chromosome number. So for example, if this is chromosome 4, this is also chromosome 4. However, they are not identical. One is from your dad. You inherited, for instance, this chromosome 4 on the left from your dad, and you may have inherited this chromosome 4 from your mom. Obviously, your mom and your dad should have different genes, or at least different alleles of a gene, but the same types of genes would appear on these chromosomes. So again, what do you need to know? You need to know that homologous chromosomes are not identical. One was inherited by your mom, and one was inherited by your dad. However, sister chromatids, you should know that these are identical copies, right? Sister chromatids are identical copies of a chromosome, like these two red ones here. It's like if you took this red one and copied it, you'd have two red ones. They're identical in every way. Okay, so please know the difference between homologous chromosomes and sister chromatids. And again, this video right here sums it up nicely for you. All right, next, what else do we need to know? We need to know that the cell cycle consists of two major phases. Mitotic phase, also known as M phase, and interphase. Okay, so what is mitotic phase? Mitotic phase, or M phase, consists of mitosis, and also it consists of cytokinesis. Remember that cytokinesis is not part of mitosis. Cytokinesis and mitosis are part of M phase. This is when the cell is actively dividing. Mitotic phase is where the cell is actively dividing. The other part of the cell cycle is known as interphase, and this is the part of the cell cycle where the cell is growing and copying its chromosomes. Let's break down interphase, and then we'll break down M phase. During interphase, this takes up 90% of the cell cycle. So please know again that there is interphase and there's M phase, right? And know that M phase consists of mitosis and cytokinesis. Okay, but cytokinesis is not part of mitosis. Right? Now, we're breaking down interphase. We need to know the three subphases of interphase. What interphase, first of all, remember it takes up 90% of the cell cycle. So it takes up most of the cell cycle. 90% of cell cycle. We need to know that. What else should we know about interphase? We need to know that there are three parts to interphase: G1, S, and G2. And we're going to break down what happens during G1 of interphase, what happens during S, and what happens during G2. We're going to break that down. So here you can see the cell cycle. Remember, you have, this is interphase here, the yellow arrow, the orange arrow, and the green arrow. These constitute interphase. And then you have the purple and the blue arrows, which comprise M phase, or mitotic phase. So let's break these down. M phase, remember, M phase is made up of mitosis and cytokinesis. Now, mitosis is further broken down into subphases. Remember, mitosis is broken up into prophase, prometaphase, metaphase, anaphase, and telophase. We do want to know these phases of mitosis, and that's followed by cytokinesis. So let's go through one by one. Interphase. Okay, what do we need to know about interphase that I want you to know for the exam? Interphase. Remember, interphase is 90% of the cell cycle. We need to know that it's most of the cell cycle. It takes the longest time. We need to know that interphase is made up of G1, S, and G2 subphases. So what happens during G1? We need to know that. During G1, cells grow. Cells grow. What happens during S phase of interphase? DNA is replicated. Yeah, you copy the DNA. And this is when you end up with your sister chromatids, by the way. This is when you end up with your sister chromatids. And by the way, if you want a nice video over interphase, click on this link at the bottom left. Then we have G2. What happens during G2? We need to know that during G2, the chromosomes coil up. The chromosomes coil. And what else do we need to know? The centrioles replicate. These are your centrioles here. The centrioles replicate. Great. That's all we need to know about interphase. Let's move on to M phase. Remember, M phase or mitotic phase starts with prophase. Prophase. What happens during prophase of mitosis? What I need you to know is this. During prophase, sister chromatid pairs form and pair up. Sister chromatids, remember the two identical copies of all the chromosomes? Sister chromatids form and pair up. Okay, that's what these little X's are, these blue and red X's. Prometaphase, what do we need to know about prometaphase? During prometaphase, the nucleus breaks down, the nuclear envelope breaks down. Okay, you see that? Nuclear envelope breaks down, and the sister chromatids move or start moving to the center of the cell. So you see here the nuclear envelope. Here there was a nucleus, right, in the nuclear envelope. Here in prometaphase, the nuclear envelope has broken down. Microtubules are attached to the sister chromatid pairs. And notice how the sister chromatids are moving to the center of the cell. What do we need to know about metaphase? What should you know about metaphase for the exam? Metaphase, look what's happened during metaphase. You see this? What's lined up at the center of the cell? It looks like the sister chromatids, the sister chromatid pairs lined up down the center of the cell. Sister chromatid pairs lined up down the center of the cell. Then what happens during anaphase? Anaphase, look at this. Sister chromatids separate from one another. Sister chromatids separate from one another. Then telophase, the daughter nuclei form. A nucleus forms here and a nucleus forms here. So daughter nuclei form. And that's followed by cytokinesis, right? Telophase is followed by cytokinesis. And remember, how does cytokinesis occur in animal cells? In animal cells on the left, cytokinesis occurs by cleavage furrow. Animal cells, you have cleavage furrow. That's how the cell divides. or at least that's how the cell itself divides during cytokinesis. Remember, the cytoplasm is split when the pinching of the membrane occurs with cleavage furrow. In plants, however, we don't have a cleavage furrow, do we? Remember, in plants, cytokinesis occurs by cell plate formation, cell plate. So in plants, you need to know that in plants, cytokinesis occurs by this one, a cell plate forms. Whereas in animal cells, cytokinesis occurs by cleavage furrow. Awesome. What else do we need to know for the exam? One thing I want you to know real quick is that what is the result of mitosis? I need you to know this for the exam. Mitosis results in what? Mitosis results in two cells, right? Two cells. But you need to know it's not just two cells, it's two genetically identical cells. Does that make sense? You had 46 chromosomes, you copied all the chromosomes, they paired up as sisters, and then they separated down the center of the cell. This is going to result in two genetically identical cells. Okay, so I want you to know that mitosis results in two genetically identical cells. Here it is. See, oops. Two genetically identical daughter cells. Now, binary fission we can skip for the purposes of this class, and we can also skip the cell cycle control mechanisms at the end. All right, so let's stick to those topics that we just covered together, and I think you'll be just fine for the exam. Please note the different subphases of M phase and interphase. I think we'll be set. And again, remember that mitosis results in two genetically identical cells. And mitosis results in the somatic cells. All right. Well, I hope this review was beneficial to you. Please focus on these things that we touched on during this review video. I wish you well. I know you can do it. Study hard. And I wish you the best of luck on the exam. Continue the hard work. And let's finish strong.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.